In this video we will discuss about effect of ECM stiffness on angiogenesis, drug resistance, immune escape and metabolic reprogramming in brief. So we will start with this effect of ECM stiffness on angiogenesis. In the previous video we have understood that the ECM stiffness helps in this angiogenesis formations and that helps the food supply to reach to the tumor cells. Apart from that, this angiogenesis also helps in this endothelial cells proliferations by increasing the formation of this VEGF, which can interact with this VEGF receptors on endothelial cells. On the contrary, this ECM stiffness will prevent the endothelial cells migrations by providing physical barrier as a through this increase in the stiffness, and also it will release this anti-angiogenic factor through ECM fragmented proteins. And so this is about the angiogenesis in brief, how it will affect the endothelial cells positively and negatively. And now about the drug resistance. So when it comes to the drug resistance, what it does is increase in the stiffness helps in the upregulation of this MRC1 ABC transporter, and we have discussed this thing in detail, how this ABC transporter helps in the removing the chemoresistance drug from the cytoplasm or excluding the chemoresistance drug from the cytoplasm of the tumor cells. So matrix stiffness, what it does is it increase the ABC transporter MRC1 in the tumor cells, and that will prevent this or prevent drug resistance, or this leads to the drug elimination. At the same time, it will also prevent the T cells interactions with this antigen presenting cell surface epitopes on the tumor cells. And when it comes to the immune escape, the T cell interactions with the immune cells also plays an important role in this immune cell escape, that this ECM, because of its physical barrier, prevents the T cells interactions with this anti-APCs that are present on the tumor cells. Anti-APCs means antigen presenting cell surface, that helps the tumor cells to detect that these are not good cells for the body. So this and at the same time, what it does is it will leads to this YAP-TAZ nuclear localizations, and that will cause epigenetic changes in the cells in a way that will leads to the TME compositions changes towards forming more myeloid derived stem cells, and that will, and also they will form this immune checkpoint checkpoint receptors that will inhibit the immune cells through, for example, this anti-CLA4 or this anti-PD1. And at the same time, so this is about the immune escape. And now it comes to this metabolic reprogramming. So the three pathways that are mentioned here is about this ECM stiffness regulate the glycolysis pathway, lipid metabolism pathway, and also this amino acid metabolism pathways. And this regulations occurs in a way that will help the tumor progression. For example, it will leads to it will change the glycolysis pathways in a way that most of the energy consumptions are now being consumed for the cell growth rate and cell proliferations. Cell growth and cell proliferations without utilizing the energy for other pathways that are not necessary for the tumor cells survival. And at the same time, there is amino acid pathways such as this arginine will go down, and this decrease in the arginine will help the cells in a way that it will cause microtubule stability in the newly changed status of these tumor cells. And so this is, and last one is the lipid metabolism pathways. So they will also interacts. This will also act as a physical barrier in a way that will prevent, or that will helps in the immune evasions. So that's all for this video. I hope you understood how this stiffness regulate angiogenesis, drug resistance, immune escape, metabolic reprogramming in brief. If you need to understand more about this topic in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.